So, good afternoon, morning, evening, night, pick yours. So in this validation and remediation session number 7, we will hear 4 talks. Well, hopefully, so far there are only 3 speakers present from colleagues who really, in my opinion, do not need much introduction, so I can probably stop stealing their precious time. And if nobody knows them, then you can actually read it in the Slack or in the web. So just really quickly, Robbie will tell us something about the recent development in PDB Redo. Then if Jane appears, she will tell us and probably show us why and how we need to introduce new validation approaches. John will show us that glycans indeed do matter, and Gerard will conclude with giving us some guide into this post-AlphaFold world. So everybody knows how it works. You have each 20 minutes, 15 minutes are kind of supposed to be for the talk, 5 minutes for discussion. So I'll signal 15 minutes. And with that said, Robbie, floor is yours.
Thank you. I'm going to talk about using PDB Redo as a data source for refinements, validation, and more. So in the PDB Redo project, we make tools for structural biology, and we focus not just on the protein, but also nucleic acids, ligands, sugars, and metals. And our goal is to help people with their research by creating tools to analyze models, but also to make new programs and algorithms to make better structure models. And we also provide updated structure models for analysis and data mining. At the core of the PDB Redo project is the PDB Redo pipeline, and that's a fully automated pipeline that refines, rebuilds, and validates your structure. And it uses software from CCP4 like RefMac and Qt, but also a lot of tools that we have developed ourselves. PDB Redo comes in 2 flavors, both of them available on pdbredo.eu. On the one hand, we have a web server where you can use the pipeline to redo your own structure. But we also have a data bank of redone PDB entries, and there's 150,000 of them at the moment. And this is a setup with positive feedback, because the new methods feed the data bank, but at the same time, we can use the data bank entries to feed model development. And I want to focus on the latter. So why would you want to use PDB Redo as a data source? Well, first of all, the models are updated with new methods. And that's important because PDB models were made with the methods of their era. And the PDB has been around now for half a decade. That means that some of the entries are actually really old. Also, the models in the data bank are generally of improved quality and have fewer errors. So here we see an example below, where in the PDB Redo model, after some refinement and sidechain rebuilding, we can see a better perspective on the UDP binding mode, and we can see that the base is sandwiched between 2 aromatic rings. Of course, because the method is automated, we also do sometimes introduce errors in the structure models. What is also important is that it's a consistent procedure for all models. So that sort of wipes the memory of structure models being done by different people with different software. And this consistency is particularly important for data mining. We also know we also document which restraints were used and which weren't. For instance, we never use Ramachandran restraints. And we use consistent geometric restraints. And this is important because over the years, the restraints that we use for refinement have changed, particularly for nucleic acids. And I'll come back to that later. And last but not least, we have very rich and fair metadata that can be used to filter data sets. So let's look at an example. Well, yesterday we talked a lot about restraints, and you could ask yourself, what would be a good target for hydrogen bond length? And there's different approaches to that. Well, you can do the hardcore mining thing, where you say we use time-specific restraints, and we mine them in advance, and that's what we did a few years ago. We took 10,000 non-redundant PDB Redo entries, good resolution, good fit to the experimental data. And from that, we took 4 million observed hydrogen bonds, and we fitted targets for 118 different types of hydrogen bonds, so with different donors and acceptors and secondary structure. That sort of worked, but lately we use a new method that uses context-specific restraints, and these are mined on the fly because you cannot prepare that beforehand. So what I mean with context-specific is that for every hydrogen bond in your model of interest, we actually look at other observations of the similar hydrogen bond. So we take all the homologs, at least the close ones, and we look for the equivalent hydrogen bond, and then we fit a target on that. Now you want to make sure that these restraints are as good as possible. So you need to filter on the quality of the models. And we do that by using the real space correlation coefficient. Now, in some cases, there are lots and lots of homologs, hundreds or even more. Then calculating the real space correlation coefficient for everything takes ages. So that's why we have this information already available, and we can do that very quickly. Different projects involved benchmarking carbohydrate restraints, and this is something that we did together with John Aguirre and also with Rob Nichols. And what they did is they came with new restraints for pyranoses, so that's sugars. And those were made with ACE drug, and then they were patched with unimodal ring torsion restraints. Now the question is, are these restraints actually an improvement? Well, to figure that out, you need to do very careful refinement, changing only the different sets of restraints. But of course, if you have a test set, then you have different structures with different data qualities and different resolutions. So for each one, you actually first need to figure out how to refine them. And that takes a long time. But in our case, we could just get the optimized refinement parameters from the PDB-Redo data bank and then use that for the benchmark. So that's what we did. We ran 3 different RefMech refinements on 960 structures. And with that, we could show that the new restraints themselves already take out some of the distorted rings in and glycans here on the right. But if you actually switch on these torsion restraints, you can get rid of even more distortions. At the same time, you want to be careful that you don't actually fix true outliers. And we showed that that is actually the case. So here you have a sialic acid that has a non-standard ring conformation, but it's very well supported by the experimental data. And that's why these torsion restraints don't actually make the structure any worse. Also, this goes for other structure quality metrics. Those are not hampered by these new restraints. So the answer is yes, these new restraints are an improvement. We also developed some other restraints recently, and those were Watson-Crick base pair hydrogen bond restraints. And here, instead of mining the PDB-Redo data bank directly, we first updated all the nucleic acid containing entries because there were new restraints available, and those were from Gilsky et al. Published a few years ago. So we first wanted to improve the uniformity of the data set. And after that, we could do the same trick with data mining, taking high resolution structures and cases with good fit to the experimental data. In doing so, we came up with 17 case-specific distance restraints for different types of base pairs. But now the question is, how do you actually test the value of these restraints? Well, yesterday you heard about DNATCO, which is a very good validation tool for nucleic acids, but it specializes in the backbone and not so much in base pairs. So we needed a new independent base pair validation system. And to do that, we looked at other parameters of base pairs, and particularly the relative orientation of the bases. So those can move with respect to each other in both rotation and in translation. And then you get these parameters called buckle, propeller, shear, and stretch. We mined that from the same data set as before. And that gives us distributions for all parameters and all the different types of base pairs. So now you can assign a Z-score for each base pair parameter. Or if you want to look at whole structures, you can calculate an RMS Z-score for that. But in some cases, you don't want 4 scores, but you want 1 overall score. So we also created a combined Z-BPG score, or an RMS Z equivalent. So with this validation now in hand, we could finally test our hydrogen bond restraints. And we implemented them in PDB-Redo in combination with the base pair stacking restraints from libg. And we tested it over 6,000 structure models. And what we saw is that in the majority of cases, you have better fit with the experimental data. Clash score improves a lot. That's always good. There was no obvious trend in Cornfile score, so things didn't get any better, but also not any worse. Of course, the hydrogen bonds themselves become much more normal, but that's something that you restrain, so that's not a surprise at all. But we see that there's a general improvement in base pair quality. And in the plots below, you can see that most observations are either on or below the diagonal. And the ones in blue are actually significant changes. So these restraints work quite well. Here I show you an example of the mu transpososome. And you can see that there's certain base pairs that are quite heavily distorted, and that's why they're marked in red. And there's an example, a base pair below, where you can see that the bases are actually in the correct orientation, but they're just way too far apart to actually form hydrogen bonds. So this isn't a real base pair then. Look at the PDB redo structure, you can see that the distortions have gone away, and now we have perfectly nice base pairing as well. So this is another approach, but let's look at something completely different now. So during this meeting and during a lot of meetings in the last year, people have talked a lot about AlphaFold, and let's continue with that. So AlphaFold has been very successful in predicting the structure of proteins, which is very good news. The problem is that proteins alone do not tell the whole biological story. So here on the bottom right, you have the structure of human myoglobin. But without the heme, that doesn't really tell your biological story. And this is the case with all the AlphaFold models, the ligands and such are missing. But at the same time, the local binding site for these ligands are really well predicted. So that leaves us with the question, can we enrich AlphaFold models with ligands and cofactors? And the answer is, yes, we can. So we made a new tool called AlphaFill, and that finds ligands and cofactors for these AlphaFold models. Based on a selection of 400 compounds, so that's the important ions, the important cofactors, but also all the different analogs that are used in crystallography to capture a protein in a certain state. And the algorithm works as follows. So for every AlphaFold model, you blast against PDB redo databank. You take all the hits sorted by E-value, and we only take the hits with 35% sequence identity that have high scoring pairs of at least 85 residues. We do a global alignment to get the RMSD of the hits, but the more important part is for the compounds themselves. So we go through all the hits, and for each compound in the hits, we do a local structural alignment with only the atoms around the binding site. And then we transplant the compound if there's not an equivalent compound within 3.5 angstroms. So that means that if you have 50 homologs, of which 10 have ATP, we only fit 1 ATP. But we also map compounds to the canonical form. So if you have a structure with AMP, PNP, which is a known ATP analog, we change that into ATP by changing atom names and, if necessary, also deleting atoms. So we applied this method to the AlphaFold databank, and we got 365,000 AlphaFill models, of which 95,000 actually have transplanted compounds. And in total, we transplanted some 830,000 compounds. Of course, you want to have some sort of validation on this, and for that, we looked at the local and global alignment RMSD. So if you look at the distribution of RMSDs for high confidence transplants, so things with 70 to 100% sequence identity, anything that is an outlier in there, so outside of the whiskers of the box plot, we call suspect. And if we take all the transplants that we did and any outliers there with respect to the whiskers are considered unreliable. And using these criteria, then we can say that 62% of the transplants is high confidence, 90% is suspect, and 18% is deemed unreliable. That doesn't necessarily mean that they're wrong, but it means that you have to be extra careful. So for example, here we've got our myoglobin again, and you could see that in the AlphaFill model, different compounds are placed. And if we zoom into the heme binding site, you can see heme, but you also can see an oxygen molecule and 2 carbon monoxide molecules. And you can see that one is on apparently the wrong side of the heme, but that is actually experimentally observed in one of the structures that we use as a parent for this. There's another example of different zinc binding sites. And on the top left, you see a functional zinc site, and the other ones are all structural zinc sites, except for the one on the bottom right. That one is from the zinc finger protein 91, which doesn't have any close homologs in PDB, only distant ones. And you can see that this transplant is not particularly good. This is not a zinc site. But the good thing is that this one is also actually marked as unreliable. So that's why these metrics actually matter. Now, if you want to use AlphaFill, you just go to alphafill.eu, and you can search on UniProt primary accession code. That's the same code as AlphaFold uses. And you can get entry pages like this one. And on the right, you have a table with all the different transplants. And if something is suspect or unreliable, it's marked in yellow or red. And you can toggle off individual transplants. Or if you click a line, you can zoom into that in this Molstar viewer. You can also filter the transplants based on identity. And of course, download the model in MMSIF format and the metadata. Now, if you want to have all of them, you can also get the whole data bank through rsync. And as you know, in December, there was a new release of AlphaFold DB. And we're processing that one, and it should be available next week. And then we'll have more than 800,000 models with transplants. So to sum things up, you can use PDB Redo and the metadata for different applications. You can use it to make restraints. You can use it to optimize refinement parameters. You can use it to create data sets for validation, or you can use it to purely fill purely computational models. And of course, I don't do that alone. We have a very good team working on that and some important collaborations. Here I want to highlight Ida de Vries and Maarten Hekkelman, who have done all the work for the AlphaFill project. But I also want to mention our collaboration with PDBE and with the MRC, Garib, Paul, Rob, Faye, and for the Sugar project, John Aguirre and Michaela Atasadova. Funding comes from these sources below, and here I want to particularly thank CCP4 for their support. And with that, I finish, and I hope there's still time for questions.
Yeah, the timing was almost perfect. I seen acknowledgement, so I didn't have to really call the 15 minutes. It was perfectly okay. We have probably like 2 minutes for discussion. There is one comment that people like your nice background. I already seen it, but it is really good.
That's always good.
So back to relevant questions. In Slacks, Rene is asking if AlphaFill could be also used to find sites for post-translation modifications. I guess it can in principle.
Yes, that could. Although one of the things that post-translational modifications do is that they actually change the structure substantially. So if you look at phosphorylation in kinases, that changes loop conformations and that sort of thing. So it's something that you can do. But there's a bit more risk involved in getting the structure wrong. And the problem with AlphaFold is that the actual biological state that they model is unknown. And this is particularly important for structures that have large conformational changes.
That's right. It would probably deserve like more detailed discussion. Because actually the model can include the effect of the post-translation modification already. So yeah, it might be actually like unmodified might be wrong.
Yeah, you don't know. You have to figure that out. And that's part of the challenge.
We have a question in the Q&A, and that's from Gerard Brickham. And the question is, wouldn't the AlphaFold predictions be biased towards allowing space for large missing cofactors just because its learning was done from structures that contain them?
I don't know if they're biased, but so I don't think there will be ghost sites there. That is something that you should be able to figure out, but then you have to do a sort of prediction of binding sites and docking of things without the context of homology. It's an interesting question. I don't know the answer, but I don't expect that to be true, but it might actually be the case. And the other question is, can I upload my predicted model? Not yet, but there will be something for that at some point. We'll make a web service out of this as well.
Okay, thank you, Robbie. I'll probably ask you if you can switch to Slack and answer the remaining question that's there.